Nervous system embryology starts off the same way as all embryology, and that is, of course, when the sperm and the egg come together to form a single cell we call a zygote. Now, the zygote's goal is to travel to the uterus so it can have a good environment to develop into a full baby. But on its way to the uterus via the fallopian tubes, it undergoes mitosis or cell replication quite a number of times until it ends up forming a cluster of cells we call a morula. The morula then develops a cavity on the inside of it, as shown here, and we call that new structure with the cavity a blastocyst. Once the blastocyst is formed, it will then implant into the uterine wall and undergo a process called gastrulation. Gastrulation is a big process, but essentially it results in the differentiation of these cells here into three main categories, and they are the ectoderm, the mesoderm, and the endoderm. We also have a black structure in the middle here called the notochord that comes from your mesoderm, and essentially it helps orchestrate the different processes we'll go through. So let's take a cross-section of our gastrula, and as you can see here, I've just taken out the ectoderm. The reason why I've taken out the ectoderm is because your entire nervous system is sourced from your ectoderm. So whilst all of these other structures are still here, we're just focusing on the ectoderm for simplicity. Okay, so the first thing that happens is the notochord, which is the black structure in the middle, stimulates the ectoderm to both thicken and also differentiate some of its cells. So you can see here, the ectoderm has thickened, and we also have some new cells here in different colours. In the middle, in the green, we have the neural plate, and on either side of the neural plate, we have the neural folds, which are shown in black. Once those form, the neural folds will then slowly pinch together, and as you can see, once they pinch together, the neural plate begins to sag. We end up forming a new structure called the neural groove. Once the neural groove is formed, the neural folds will then come even closer together until they pinch, and the neural folds will actually pinch off. As you can see here in black, they've pinched off. They end up forming a new structure called the neural crest, and that's the source of all of your peripheral nervous system and many other structures from other systems. The neural groove ends up doing something very similar. Once it makes contact, it peels off the main ectoderm structure, and we end up forming a structure called the neural tube. That's going to be the source of your central nervous system. So before we move on, I just want to take a dorsal view of this same process. What I mean by a dorsal view is it's as if we're cutting the gastrula here and looking down. I find it quite hard to link the cross-section to the dorsal view and get my head around that. So what I've found has helped is to get a piece of paper. So far we've been taking a cross-sectional view like this. The cells have differentiated, the neural folds are coming together, forming the neural groove, pinching off, forming the neural tube. Now we're looking this way. So we're looking at this. This is what we see at the dorsal view. So it's as if we're standing in the ectoderm and looking down at this structure here. This is what we would see. Okay, so we get our cells differentiating. We get the neural plate, the neural folds. This is the important part, and this is why we're taking a dorsal view. You can see here that when it's pinching together, it's actually pinching in the middle first. So it's not all coming together at the same time. It's actually coming together in the middle, and you can see here we have an opening at the top and the bottom. So we have an opening at the cranial and caudal ends. So you can see here, we named them the anterior neuropore, the one that's cranial, and we call the bottom one caudal. We call it a posterior neuropore. So once they form, we then get closure of both neuropores at the same time. So in summary, the cells differentiate, the neural groove forms, ends up clicking together in the middle first, and then sealing both neuropores at the same time. So we end up with an enclosed neural tube. So how does the neural tube end up forming the different CNS structures? So the tube is all ready to go. It then develops three lumps: the prosencephalon, the mesencephalon, and the rhombencephalon. The pro and the rhomben will split into two structures. Both of them will split into two. So the prosencephalon will develop into the telencephalon, this Mickey Mouse looking structure up here, and the diencephalon, which is just below it. The rhombencephalon, the bottom lump, will split into the metencephalon and the myelencephalon. The mesencephalon is a little bit weird because it doesn't actually split at all. It stays as the mesencephalon. So you can see here, it doesn't change, and we call it the same thing. Now the important part to remember about this structure is that it came from a tube. meaning that the inside of it is going to be hollow. So it has walls, but a hollow inside. So you can see here, the walls of this structure will develop into the structures of your CNS. But the inside of it, the hollow parts, will develop into your ventricles, because after all, ventricles are just empty space full of fluid. So the telencephalon, the inside of it, the cavity on the inside, will form your lateral ventricles. The diencephalon, the cavities of the diencephalon will form your third ventricle. Then we have our cerebral aqueduct, and we have our fourth ventricle. Okay, so those are our ventricles. What about the walls? Our telencephalon will form our cerebrum. Our diencephalon will form our thalamus, hypothalamus, epithalamus. And our mesencephalon will form our midbrain. And lastly, the metencephalon will form the pons and the cerebellum. And the myelencephalon, the medulla oblongata. So that's CNS embryology, but why is this important and why do we need to know this? It's mainly about this part here, where the neuropores are closing. If that doesn't happen, we can end up with some really severe illnesses that are often lethal. So if your anterior neuropore doesn't close, we end up with problems at the top of our CNS and development of those structures. So you can see here, it's our cerebrum. So you can end up with an issue called anencephaly, which is basically the absence of a good part of your cerebrum, the top of your skull and the top of your scalp. Looks like a honestly horrible condition, and yeah, it is usually lethal. Another condition you can get is encephalocele, which is like an outpouching of the occipital part of your skull. That can be a problem because different structures can herniate and we're not getting proper closure of a lot of structures, so that can be quite debilitating as well. If your posterior neuropore doesn't close, we end up with a condition called Spina Bifida, which is basically similar to encephalocele, just in a different location. So our posterior neuropore is at the bottom, or the caudal end, isn't it? So we end up with structures at the spinal cord having problems. So our spinal cord can end up not closing properly, and we end up with an outpouching at the bottom of it, causing different structures to herniate. So yeah, that's nervous system embryology, at least my understanding of it.